Hello बच्चों, कैसे हो तुम लोग? I hope every one of you are doing very well and all of you are studying very hard. Okay, so today's topic is biotechnology. Actually, the basics of biotechnology will be discussed in today's video. So let's see what are the different topics that I'll cover today. The first of all, I'll discuss about the definition of biotechnology. Biotechnology actually किसे कहते हैं? Next, the types of biotechnology. Definition of recombinant DNA technology. Actually, recombinant DNA technology और genetic engineering किसे कहते हैं वो डिस्कस करेंगे नेक्स्ट. उसके बाद definition of molecular cloning and last of all the major discoveries, the discoveries that led to the development of recombinant DNA technology or modern biotechnology. कौन सा ऐसा discoveries था जिसके लिए biotechnology possible हुआ? Okay, so these are the topics of today's discussion. But before going to the detailed discussion, I'll request one thing, that please subscribe this channel and like this video to encourage us to make more and more videos for all of you. And another request I'll give here, that please take it as a class, not as a video. Watch this video till the end, and if any doubt arises, please write in the comment box and I'll definitely try to answer at the end of the video. Okay, so let's start. So what is the meaning of the word biotechnology? Biotechnology actually किसे कहते हैं? So the very basic definition of biotechnology is that utilization of any biological entity. Biological entity means that it may be microorganism or any cell of higher organisms. Okay, or any components or constituents obtained from these organisms, either microorganisms or the cells of higher organisms. वहाँ से अगर कोई components, cell का कोई component like enzyme और protein भी अगर obtain करते हैं, वहाँ से मिलता है, तो उस चीज को ऐसे इस्तेमाल करना, use in such a way that some useful product or any service is generated to enhance the human lifestyle. Okay, so any microorganism or any cell of the higher organism or any component obtained from them is used in such a way so that any product or कोई भी service generate हो जो human lifestyle को और human benefit में use हो. Okay, so उसे ही हम लोग biotechnology कहेंगे. Next, though the term biotechnology is originated very recently in recent years, but the biotechnology is not recent. It is actually used from hundreds of years ago. Humans are utilizing or getting benefits from biotechnology from hundreds of years ago. For example, you can see here that before 6000 before Christ, yeasts have been used by humans to make wine or beer. Or yeasts are also used for making leavened bread by human being before 4000 before Christ. Okay, so utilization of microorganisms as told in the definition itself. So these are also the utilization of microorganisms for human benefit. So these are also biotechnology. Or another best example is that you use yeast cells also to make curd in the kitchen. Okay, so this is not a new thing. People are using microorganisms from hundreds of years ago for making different things. So all these are also part of biotechnology, but these are known as old biotechnology. Okay, so any kind of application of microorganisms for human benefit by using the natural abilities of the microorganisms is considered as old biotechnology. Microorganisms का जो natural ability है, उसको utilize करके अगर कोई human benefit होता है, तो उसको हम लोग बोलेंगे old biotechnology. And if the microorganisms have been genetically modified for enhancement of their production of their product or the service, then we'll call it as a new biotechnology. So genetic modification have been done. It is possible because of the development of genetic engineering. Okay, so genetic engineering जब discover हुआ mainly 1972 or 1970s के बाद 1972-73 or after the 1970s, actually, the microorganisms have been genetically modified for their enhanced application for any production of any product or generation of some service. So these are few examples where the microorganisms have been genetically modified. For example, making of human food from fungal origin. Marketing was done in 1980. Okay, the use of monoclonal and clonal antibodies, USA approved that in 1981. Approval of the use of insulin produced by genetically engineered microbes in 1983. Okay, animal interferons have been produced by genetically engineered microbes and approved in the protection of cattle against diseases in 1984, and many other things like production of genetically modified crop plants like flavor saver tomato or any other crop plants have been done in 1990s. So all these which are regarded in which are presented with a red colored font is considered as new biotechnology or only biotechnology. Okay, so actually बायोटेक्नोलॉजी जो कहते हैं हम लोग अभी रिसेंटली जो बायोटेक्नोलॉजी मीन करते हैं वो applications of genetically modified microorganisms but not the natural abilities of microorganisms. Okay, so these are the two categories of biotechnology we can categorize the biotechnology in two different ways. Okay, now let's come to the modern definition of biotechnology. What is the why modern definition is required? Because nowadays many processes we have seen in the basic definition that any kind of applications of microorganisms or their product will be regarded as biotechnology. But many processes are there in recent times in which neither the microorganisms are used or nor the products obtained from them are used, but they are regarded as biotechnology. For example, the in vitro fertilization process. All of you know that the pears who infertile pears, particularly who face different problems during their reproduction or giving birth to their child, they carry out in vitro fertilization of their sperms and eggs to generate fertilized embryo, and that is implanted into mother's womb to give birth of a test tube baby that we call test tube baby. Okay, similarly synthesizing any gene, कोई भी जीन डीएनए सीक्वेंस को आर्टिफिशियली सिंथेसाइज करके उसको यूज किया जा सकता है. नहीं तो डेवलपिंग ऑफ डीएनए वैक्सीन्स के डेवलप करने के लिए कोई भी माइक्रो ऑर्गेनिजम्स का ज़रूरत नहीं है. सिर्फ डीएनए सीक्वेंस अगर पता हो तो वो डीएनए सीक्वेंस को आर्टिफिशियली सिंथेसाइज करके उसको यूज़ किया जा सकता है. सो दीज एप्लीकेशंस डज नॉट नीड इन ए माइक्रो ऑर्गेनिजम्स, लिविंग ऑर्गेनिजम्स or in product obtained from them. So, but these are regarded as part of biotechnology. So that is why the European Federation of Biotechnology, that is EFB, has given a definition of the biotechnology that encompasses both the traditional view. Traditional view means that definition which we just discussed in earlier slide, and the modern molecular biotechnology. They combined both these views and given a definition which is the integration of natural science, means natural science and organisms ka integration. Any microorganism or cells of the higher organisms or parts thereof, means organism के cell से जो proteins या enzymes obtained होता है वो parts means that, and molecular analogs means any artificially synthesized molecular analogs. Analogs means the compounds which are similar to cellular compounds. Okay, that will be artificially synthesized but analogous to the cellular components, cellular molecules. Okay, for the products and services. So the integration of natural science and organisms, cells, parts thereof, and molecular analogs for the production of any product or any service that will enhance the human lifestyle. Okay, so I hope now both the traditional view of definition or the modern definition of biotechnology is clear to all of you now. Okay, let's see the types of biotechnology based on the fields of application. कौन सा फील्ड में बायोटेक्नोलॉजी का एप्लीकेशन होता है, उसके बेसिस पे different types of biotechnology can be there. First of all, if the biotechnology application of biotechnology happens in the field of medicines, that means for generation of any drug or any antibiotic or vaccine, so the biotechnology in that case will be known as biomedical biotechnology, or it is also known as red biotechnology. That is why I have highlighted this, written this term in red colored font. Okay, red biotechnology, keep this in mind. Medical biotechnology is red biotechnology. Okay, next type is the industrial application of biotechnology. Okay, industry means any type of industry, like making of ethanol or any organic acids, or making of any food, fermented food products. Any type of industrial application will be known as industrial biotechnology, or it is also known as white or gray biotechnology, either white or gray biotechnology. Okay, so keep these terms in your mind, that which one is called in which color. Okay, next category is agricultural biotechnology. From the name itself, you can understand that application of biotechnology in improvement of agricultural crops, rice or brinjal or cotton or any type of crop plants. If improvement is done by using biotechnological applications, then it will be known as agricultural biotechnology. Maybe the improvement may be in terms of nutritional enhancement of the nutritional value, or enhancement of their pest control ability, or any other way. So this will be regarded as green biotechnology. Okay, green biotechnology. Clear? Another category is if the biotechnology is applied in the field of improvement of marine or aquatic organisms. Okay, marine or aquatic biotechnology. It will be known as marine or aquatic biotechnology, or in terms of color, it is known as blue biotechnology. So these are the major types of biotechnology, applications of biotechnology actually. Okay, I hope all of you now understand what are the types of biotechnology. Okay, now let's come to the definition of recombinant DNA technology or genetic engineering. Recombinant DNA technology किसे कहते हैं, or genetic engineering, the same thing, but it is known in different way, different terms. Genetic engineering and RDT is the same thing. Okay, so before discussing the definition, let's understand this figure. Okay, what is recombinant DNA actually? Before knowing the recombinant DNA technology, let's learn what is recombinant DNA. Okay, so you can see here that two different DNA molecules are shown, DNA 1 and DNA 2. These two DNA are coming from two different sources. Okay, suppose, for example, this one is coming from E. coli cell. E. coli is a bacterium. Okay, and this one is coming from any mouse cell. Clear? You have isolated the DNA from bacteria, and this one you have isolated this DNA from any mice. Clear? Now you have cutted a fragment from this DNA to generate a small fragment, and also cutted this DNA from the second source to a small fragment. Now if you join these two molecules, that means DNA 1 and DNA 2, these two molecules, if you join together to make a single molecule, this molecule will be known as recombinant DNA. Okay, or chimeric DNA. All these steps, this cutting and joining, all these steps are done in vitro condition, means in test tube. Okay, so ultimately after making this recombinant DNA, it has to be introduced into a host cell. Suppose this is a host cell. Host cell means host cell may be any bacterium or any lower eukaryotes, or I'm just writing here the name of E. coli cell, because in most of the cases E. coli, that is a bacterium, all of you know, E. coli is used as the host. So this DNA, recombinant DNA, after making of it, it will be introduced within the host cell. Okay, so that this DNA can replicate itself to make more and more copies within the host cell, and is transferred and is propagated to its next generation. Okay, so this cell have obtained this DNA molecule prepared outside of the cell, and after growth of this cell will divide. So after division, 2 daughter cells will be produced. So both of these daughter cells will obtain this DNA molecule. So this is known as propagation of the DNA molecule in the next generation. Okay, so all these steps of making the recombinant DNA and to introduce it into the host cell for its propagation into the next generation is known as recombinant DNA technology. Now you come to the definition. Now you read it. That formation of new combination of heritable material. DNA is heritable material, and a new combination is formed here. New combination means taking DNA from 2 different sources, or more than 2 may also be possible. You can take DNA from more than 2 different sources and join them together. Okay, so new combination of heritable material is produced. How it is produced? Now, by insertion of the nucleic acid molecules obtained from whatever means from whatever sources, कोई भी source से different molecules obtain करके into any virus or bacterial plasmid or other vector system, कोई भी एक vector system का जरूरत होगा. For example, among these 2 molecules, for example, this one is a vector DNA. Vector means a vehicle on which vector DNA will be used as the backbone or the support on which any other gene of interest. Suppose this one is gene of interest. You want to work with this particular gene, the blue colored gene. So now this vector DNA will act as the support for integration of this foreign gene, this gene of interest. Okay, so that is why among the different DNA molecules, one of the DNA molecule will be either any viral DNA or any bacterial plasmid or other vector. Okay, and all these things are happened outside of the cell. Okay, this cutting and joining and making of this recombinant DNA happens outside of the cell, so as to allow their incorporation into a host organism. So just I have told in this particular case, so that this DNA can be incorporated into a host organism in which they do not naturally occur. They do not naturally occur means before integration of the DNA into the host cell, this combination of DNA was not there in the host cell. Okay, the host cell जो था वहाँ पे उस cell के अंदर इस combination का DNA नहीं था पहले. Okay, but after you introduce it within this host cell, in which they are capable of continued propagation, it can propagate to the next generation. पहले नहीं था, लेकिन जब ये इंट्रोड्यूस्ड हो गया तो इंट्रोडक्शन के बाद वो नेक्स्ट जनरेशन में प्रोपागेट कर पाएगा. Okay, so all these things come under the definition of recombinant DNA technology. All these steps and processes. Okay, so read again. Formation of new combination of heritable material by the insertion of nucleic acid molecules that is obtained from whatever means into any virus or bacterial plasmid or other vector system outside the cell, outside of the cell. Cell के बाहर ये सब cutting and joining होने के बाद, so that to allow their incorporation into host organism. Host organism के अंदर introduce होता है, in which they do not naturally occur, जहाँ पे वो पहले से नहीं होता है, host organism में पहले से नहीं था, but in which they are capable of continued propagation, जहाँ पे वो continuously वो propagate करेगा next generation तक. Okay, clear? So this is the basic of genetic engineering. Now let's come to the definition of molecular cloning or simply cloning. Okay, simply cloning बोलते हम लोग समझते हैं कि cloning of any or human being or any other animals or any other organism. Okay, but that is not the thing. In recombinant DNA technology, cloning means molecular cloning, that is cloning of a particular gene or cloning of a particular fragment of DNA, not a whole organism. कोई एक ह्यूमन का डायरेक्ट क्लोन बना देना, ये रिकॉम्बिनेंट डीएनए टेक्नोलॉजी के का जो मॉलिक्यूलर क्लोनिंग है वो नहीं है. ये मॉलिक्यूलर बायोलॉजी जो रिकॉम्बिनेंट डीएनए टेक्नोलॉजी में जो क्लोनिंग बोलते जो हम लोग समझते हैं वो क्लोनिंग ऑफ individual genes or small fragments of DNA. Okay, so what is the basic definition? Molecular cloning is a set of experimental methods in molecular biology that are used to assemble recombinant DNA. यहाँ पे भी recombinant DNA prepared होगा. Okay, recombinant DNA molecules and to direct their replication within host organism. Same चीज जो recombinant DNA technology में जो देखा, molecular cloning भी वो सेम थिंग है जहाँ पे न्यू कॉम्बिनेशन ऑफ डीएनए और रिकॉम्बिनेंट डीएनए विल बी प्रिपेयर्ड and will be introduced into the host cell within which it will be replicated to make higher number of copies. Means a single copy of DNA, suppose you have introduced into the host cell and many molecules of DNA will be produced. So that means the clone of the single molecule. This one was the single molecule of DNA, and from single molecule, multiple molecules of its clone. All of these molecules will be the clone of this molecule. Okay, so many molecules will be produced from a single molecule. This is why it is known as cloning of the genes or cloning of the DNA molecules or molecular cloning. Okay, so let's see again what are the different steps. Now, here also you can see that a prokaryotic cell, E. coli cell is there, within which a particular type of DNA is there. You can see here the small circles, the larger one, this is the chromosomal DNA, this large linear form, this is chromosomal DNA. Chromosome, but just ignore my handwriting because on the screen it is very difficult to write properly. Okay, so just try to understand that this larger linear form is the chromosome and all these small circles, these are known as plasmid, P-L-A-S-M-I-D, plasmid DNA. So what is plasmid DNA? All of you know, I guess, that plasmid DNA is a short and circular extra chromosomal DNA which is capable to replicate independently. Okay, जो क्रोमोसोम से बहुत ही छोटा होता है लेकिन सर्कुलर होता है and it can independently replicate. Okay, so this plasmid DNA actually are used as vector. Plasmid DNAs are usually act as vector. So you will isolate the DNA from the bacterial cell, means bacterial cell को rupture करके उसको फटा के वहाँ से plasmid DNA को निकाला जाएगा, आइसोलेशन किया जाएगा. उसके बाद ये जो रिंग लाइक स्ट्रक्चर है प्लाज्मिड डीएनए, एज आई हैव टोल्ड दैट सर्कुलर इन नेचर, जो रिंग लाइक स्ट्रक्चर है उसको एक पर्टिकुलर पोर्शन में इसको काटा जाएगा. वो कट करेगा कौन? ना रेस्ट्रिक्शन एंडोन्यूक्लियस, अ ग्रुप ऑफ एंजाइम which cuts double-stranded DNA in a specific location. So एक specific location क्या location में अगर cut कर दो यहाँ पे एक this ring-like structure, if it is cutted in a particular location, so the ring-like structure will open up just like this. Okay, so यहाँ पे opening हो गया. Clear? Now see the second source of DNA. Here it is an eukaryotic cell. From nucleus the DNA is extracted, the chromosomal DNA is extracted, and DNA is isolated and purified, and within the chromosomal DNA, suppose one fragment is there with which you want to work. यहाँ पे जो फ्रैगमेंट है, ये छोटा सा फ्रैगमेंट, इसको लेके तुम काम करना चाहते हो, डाउनस्ट्रीम कोई भी एक्सपेरिमेंट करना चाहते हो. तो इस क्रोमोसोम को, as the eukaryotic chromosomes are very larger in size, so the chromosomes are fragmented with restriction endonuclease. Restriction endonuclease, as I have already told, that restriction endonucleases are enzymes which cuts DNA in a precise location. Okay, so multiple fragments will be produced, cut karne ke baad. Okay, so now if you mix these small fragments with this plasmid DNA in presence of another enzyme, that is ligase, DNA, that is called DNA ligase. So this particular enzyme क्या करता है ये exactly opposite thing of restriction endonuclease. Restriction endonuclease DNA को कट करता है, ये DNA ligase उसको join कर देगा. तो यहाँ पे ये opening हो गया था यहाँ पे इस DNA का वेक्टर का, और यहाँ पे छोटा छोटा फ्रैगमेंट है. तो क्या होगा, ये छोटा फ्रैगमेंट ये प्लाज्मिड का जो ओपनिंग पार्ट है वहाँ पे लाइगेट हो जाएगा या ज्वाइन कर जाएगा. यहाँ पे देखो कलर को डिफरेंट यूज किया गया है. यहाँ से यहाँ तक है येलो कलर्ड, जो कि ना ये वेक्टर DNA है, और इस पोर्शन से इस जो छोटा ऑरेंज कलर का पोर्शन है, दिस पोर्शन इज द DNA from this eukaryotic cell. So this again make a circular DNA. Okay, again it makes a circular DNA. So DNA ligase actually joins these two DNA molecules to make a recombinant DNA molecule. So this one is the recombinant DNA. Clear? Now this recombinant DNA is transformed, is introduced into the host cell again. Host cell means the E. coli cell. E. coli cell. Okay, so this process of introduction of the extracellular DNA into the host cell is known as transformation. इस process को कहता है transformation. इसका एक different process है, वो बाद में किसी वीडियो में मैं डिस्कस करूंगा. ओके, डिटेल्स में. लेकिन अभी के लिए ये जान के रखो कि ये प्रोसेस को बोलता है ट्रांसफॉर्मेशन प्रोसेस. क्लियर? सो ये जो प्लाज्मिड DNA था, जो रिकॉम्बिनेंट DNA प्रोड्यूस हुआ, वो सेल के अंदर चला गया. यहाँ पे देखो मल्टीपल स्मॉल सर्कल्स आर देयर. ओके, छोटा छोटा जो सर्कुलर DNA है यहाँ पे अंदर आ गया. तो दिस बिकम्स the recombinant transformant, means the DNA is the recombinant DNA molecule, and the cell which takes up, or within which the recombinant DNA molecule is introduced, is known as transformant. जो ट्रांसफॉर्म हो गया है जिस सेल का, वो ट्रांसफॉर्मेंट के तौर पे जाना जाता है. Clear? So all these steps, starting from the isolation of DNA to making of the recombinant DNA, these are done in test tube. That means outside of the cell. ये test tube के within the test tube ये किया जाता है. Okay, restriction endonuclease apply करके digestion, उसके बाद फिर से DNA को purify करके फिर से ये T4 DNA ligase add करके उसको ligation, ये सब किया जाता है outside of the cell or in vitro. Okay, so on the other hand, after the formation of this recombinant DNA molecule, suppose only one molecule of this recombinant DNA have been produced, then after the introduction of this molecule within the cell, suppose for example this circular molecule was the original molecule, have been introduced within the cell, and within the cell it has been replicated to multiple molecules. You can count here. This is the first one, second, third, fourth, fifth. Okay, so multiple molecules have been replicated. Okay, its copy have been generated within the cell by using the cellular mechanism. So as it is happening within the cell, the replication process is happening within the cell. It is known as in vivo. Okay, within the cellular environment. So all the manipulation of the recombinant DNA molecules are performed outside of the cell, and when it is introduced within the cell it can replicate to multiple copies to generate the clones of the original molecule. That is why this process is known as molecular cloning. Okay, so next I'll request everyone to please subscribe this channel, BioSir, and like this video if you are really enjoying my teaching. Okay. Now I will discuss about the milestone discoveries that led to the development of genetic engineering or biotechnology. What were the major discoveries that actually led to the development of the field of genetic engineering or biotechnology? Okay, so first of all, I believe that the one of the foundation discovery that was made by Gregor Mendel, who actually showed that something is there which is transferred from the parent. Each parent contributes some particulate matter. Okay, some particulate matter is transferred from the parents to the offspring, and he called that as heritable substance and named the substance as elementin. The biochemical nature was unknown to him or any other scientist, but he probed by his cross-pollination experiments with pea plants that something is there which contributes to the which is transferred from the parents to the offspring and which actually determines the characteristics of the offspring plants. Okay, so this was one of the major or the foundation discoveries in the field of genetics or molecular biology or in the field of biotechnology. Okay, the next experiment that is also similar to the Mendel's experiment that was carried out by Frederick Griffith in the year 1928, when he also probed that transformation of bacterial strains of Streptococcus pneumoniae, the rough strains of Streptococcus pneumoniae can be transformed into smooth strains. Okay, rough strain of Streptococcus pneumoniae which were non-pathogenous to the mice models was transformed into smooth strains. Smooth strains were pathogenic in nature. Okay, so something was there that was transferred from the smooth strain to the rough strain and transformed them into smooth strain. So he also named that particulate matter as the transforming principle. Clear? Next experiment was carried out, and the extension of the Frederick Griffith's experiment was carried out by Avery, MacLeod, and McCurdy in 1984. They hypothesized that the genetic material is either protein or nucleic acid, because other biochemicals which are the living cells are made up of, like lipids or carbohydrates, cannot serve as the genetic material because of their chemical nature that was proven or that was known at that particular time. So either protein or nucleic acids could serve as genetic material, and they probed that the genetic material is actually the DNA, not protein or RNA. Okay, so the actual transforming principle that was proposed by Frederick Griffith, transforming principle, was proved by MacLeod and McCurdy as the DNA, not the protein. Clear? And finally, Hershey and Chase, Martha Chase and Alfred Hershey, carried out an experiment in 1952 to confirm that chemical nature of the genetic material as DNA. Okay, so I have discussed all these three experiments, that is the experiment carried out by Frederick Griffith in 1928 and the experiment of Avery, MacLeod and McCurdy in 1944 and the Hershey-Chase experiment. All these three experiments I have discussed in great detail in another video. Okay, so you can watch this video to know the details of all these three experiments. I will not elongate these experiments in this video, so you can watch from that video in my channel. Okay, so the next experiment that was carried out by George Biddle and Edward Tatum. Actually, they discovered the central dogma, that is the genes actually codes for proteins. Okay, so proteins are actually the workhorses of the cell. Genes do not perform any function at the ground level, but they code for proteins, and those proteins are actually the workhorses of the cells. Any metabolic process or any structural conformity is given by the proteins. Okay, so they propose the one gene, one enzyme hypothesis. That means each of the protein that actually functions, or each of the enzyme that actually functions during the biochemical reactions, are coded by a single gene. Okay, so this concept provided the first direct evidence that genes are actually the controlling elements of biochemical reactions. Okay, so the DNA on which the chromosomal DNA on which genes reside, those genes are actually the controlling agents of biochemical reactions. Okay, by specifying structure of the specific enzymes. Okay, so these two scientists proved that one gene equals to one enzyme. Next discovery is considered as one of the greatest discovery or greatest proposal in the field of biological science, is the deciphering of the structure of DNA molecule, that is the double helix structure of DNA, that was proposed by James Watson and Francis Crick in 1953. And I hope all of you know that both James Watson and Francis Crick obtained Nobel Prize in 1958. Okay, so they deciphered the double helical structure. All of you know that DNA is a double-stranded structure. Two strands of nucleotides are there, which remains helically wrapped around each other to form this type of structure. Okay, so that this structure was actually proposed by James Watson and Francis Crick. Okay, next discovery was made for the development of genetic engineering was made by three scientists, Werner Arber, Daniel Nathans, and Hamilton Smith. I hope all of you have understood the basic procedure of molecular cloning or genetic engineering. I have already discussed in the earlier stage of this video that to carry out molecular cloning of any particular gene, you have to cut the DNA. You have to cut the DNA at precise location. Okay, and the enzymes which actually cut DNA in precise locations are known as restriction endonucleases. Okay, restriction endonucleases. I will make another video in detail about this nature or the characteristics of these enzymes, different enzymes used in recombinant DNA technology. You can watch that video later to gain detailed information, okay, detailed knowledge about restriction endonuclease. But just in this video, you just know that these three scientists, Werner Arber, Daniel Nathans, and Hamilton Smith, discovered the restriction in endonucleases in 1970. Okay, and that allowed the DNA to be cut at specific places. Okay, they got Nobel Prize in 1978. Okay, so these were the three scientists, Werner Arber, Daniel Nathans, and Hamilton Smith. You can have a look from this figure that this was the DNA, double-stranded DNA in the first place. Okay, so hint 3 is one of the restriction endonuclease obtained from the bacterium Haemophilus influenzae. Okay, so hint 3 cuts the DNA at exactly precise location in a particular sequence. So it cuts one strand in this place and another strand in this place. So the strands are separated out from each other. So two different DNA molecules. This is one fragment, and from there, this one is another fragment. Two different fragments will be generated. Okay. Okay. So restriction endonucleases, as they cut the DNA molecules in precise locations, they are known as molecular scissors. Molecular scissors. The work of scissors, all of you know that scissors are used to cut anything. Okay. Next discovery is the DNA ligase. The enzyme actually carries out the opposite function of restriction endonucleases. DNA ligases, that is why they are known as molecular glue. Okay, DNA ligases that join broken DNA together. Okay, here two different DNA molecules are there. This is number 1. This is number 2. Two different molecules. You can see here that DNA are not linked with each other. Okay, so if you apply DNA ligase enzyme, if you mix DNA ligase enzyme with these two fragments of DNA in the same reaction mixture, this work of this enzyme is to join the DNA molecules together to form a continuous DNA molecule. Okay, so two DNA molecules were there. After joining of these two DNA molecules, a single DNA molecule, continuous DNA molecule will form. So that is why this enzyme is also known as molecular glue. Okay, for joining the DNA molecules. Clear? So actually, if you combine these two enzymes, like restriction enzymes and the DNA ligase, okay, it was possible to cut and paste the DNA sequences to create recombinant DNA. So to create recombinant DNA molecules, you have to cut DNA, and then again, two different DNA molecules coming from two different sources, you have to join those two DNA molecules together to make the recombinant DNA. So these two enzymes were the major discoveries, restriction endonuclease and DNA ligase, based on which the recombinant DNA or recombinant DNA technology or genetic engineering become possible. Okay. The next discovery was done in 1952 by Joshua Lederberg. Joshua Lederberg discovered the plasmids. Plasmids, I have already mentioned about the plasmids, that these are short and circular DNA molecules which are independent of chromosomal DNA and can replicate independently. Okay, so if you see in this figure, in bacterial cells, a chromosomal DNA is there. This one, the red colored or the maroon colored one. Okay, and here these structures, these circular structures, are the plasmid DNA. So this is also DNA, chromosomal DNA, which is much larger than plasmid DNA, and plasmids are much shorter. Okay, so these are circular, small, circular, self-replicating, double-stranded DNA. Okay, you have to remember the definition. Okay, so in definition, multiple characteristics will come, and you have to mention all of these, that the DNA molecules are small in nature, circular in nature, self-replicating. That means they can replicate independently of the chromosomal DNA, and they are always double-stranded. Okay, double-stranded DNA molecules that exist independently of the chromosome in a cell, commonly in bacteria. Mainly plasmids are found in bacteria. Okay, bacteria and archaea, and only one plasmid is found in Saccharomyces cerevisiae, yeast cell. Yeast cell, you know that yeast is an eukaryotic organism. So within eukaryotes, only yeast cell can have a plasmid. Otherwise, plasmid DNA are generally found in bacterial cells. Okay, another characteristic is that plasmids are non-essential. Non-essential means it is not essential for the survivability of the bacterial cell, but the chromosomal DNA is essential. Without the chromosomal DNA, bacterial cell is incomplete. Okay, bacterial cell cannot survive, but without plasmids, they can survive. Okay, but when present, plasmid DNAs, if they are present within the cell, they provide selective advantages to the cell. What are those advantages? Maybe antibiotic resistance, or toxin production, or metabolic enzyme genes may be there. Okay, so plasmid DNA actually contains some genes which gives some advantage to the cell. Okay, and what is the need of plasmids in recombinant DNA technology or biotechnology? Plasmids are widely used as vectors, as I have already discussed in the molecular cloning steps, that plasmid DNA are used as vectors, or the backbones on which the DNA to be cloned is inserted first to make the recombinant DNA. Okay, so to transfer foreign genes into host cells for cloning or gene expression or genetic engineering purposes. Okay, so plasmids are mainly used as vectors in recombinant DNA technology. Clear? So without plasmids, cloning would not have been possible. Okay, next discovery was done by Paul Berg in 1972. He used restriction endonucleases and DNA ligase. Both the enzymes were discovered prior to his experiment. Okay, so he used both these enzymes to create the first recombinant DNA molecule. Okay, so first recombinant DNA molecule was produced by Paul Berg in an experiment where he combined the DNA from monkey virus SV40. SV40, I guess you know that SV40 is the simian virus 40. Simian virus means the monkey virus. It is a virus, its name is SV40, with that of lambda virus. Lambda virus is a virus which infects E. coli cell. E. coli is a bacteria. E. coli cell को infect करता है lambda virus, and SV40 virus infect करता है monkey को. Okay, so the DNA from both these viruses were cutted, just like just shown in this particular figure, that SV40 DNA was there and lambda virus DNA was there. He cutted both this DNA by using restriction endonucleases, okay, and obtained small fragments, and then he joined by using DNA ligase to make the first recombinant DNA. Okay. So this was another major discovery in the line of development of genetic engineering. The next major discovery was done by Herbert Boyer and Stanley Cohen. Okay, so actually Stanley Cohen was researching with plasmids. His topic of research was with plasmids, and Herbert Boyer's topic of experiments or research was with restriction endonucleases. Okay, so they actually recognized their complementary nature of their work and teamed up or collaborated in 1972, and they actually created the first genetically modified organism. How they have created? Now, they took a plasmid they took, which is PSC101. Plasmids are named like this. Okay, so hundreds or thousands of different type of plasmids are there. Each of those plasmids are named like this. Okay, PSC101 is one of the plasmid that was the material with which the Cohen was researching. Okay, and they cut the PSC101 with restriction enzyme and inserted a gene which conferred the resistance against kanamycin. You know that kanamycin is an antibiotic against which some bacteria are resistant because they have resistance gene. So kanamycin resistance gene was taken from E. coli cell, and PSC101, this plasmid, was taken from another bacterium. Okay, and they ligated these two DNA, the plasmid and this kanamycin resistance gene together, and they introduced the whole recombinant DNA construct into E. coli cell. So that E. coli cell become antibiotic resistant. Okay, without having that gene, the E. coli cell was kanamycin sensitive, but when they are transformed with this particular construct, they become kanamycin resistant. So that E. coli cell represented the first genetically modified organism. Okay, so here you have to remember that first recombinant DNA was prepared or produced by Paul Berg, and first genetically modified organism was made by Herbert Boyer and Stanley Cohen. Okay, next major discovery was done by Morton Mendel, who produced the method to make E. coli cell artificially competent. Now, you know what is the meaning of competence? Competence means a bacteria which is able to take up external DNA. Suppose this is one of the bacterial cell like E. coli cell. Now, within environment or within test tube, a plasmid DNA or any other DNA, external environmental DNA is there. So, not all bacteria can take up the DNA inside of these cells. Okay, bacteria does not have that ability. Okay, not all bacteria can take up. Only those bacteria which are competent or physiologically active in a state in which they can take up the DNA, that physiological state is known as competence. Okay, and the bacteria can be made artificially competent if they are treated with some chemical agent like calcium chloride in 100 millimolar concentration. 100 millimolar concentration of calcium chloride, if you mixed bacterial cells with calcium chloride and treat for some time, then the bacterial cell becomes competent in nature. So after treatment with calcium chloride, they can take up the DNA. So this process was discovered by Morton Mendel, who made E. coli cell artificially competent. Okay. Later, 2 years later, Stanley Cohen also showed that calcium chloride treatment was also effective for uptake of plasmid DNA. Okay, so Stanley Cohen also carried out the similar calcium chloride treatment of E. coli cells, in which E. coli cells become competent to take up plasmid DNA within it. Okay, so this was another discovery by which bacterial cells could be transformed, or the recombinant DNA could be introduced within the host cell. Otherwise, the genetic engineering would not have been possible. Okay. Okay, so this ability actually, okay, let's see this particular definition of the word competence: the ability of bacterial cell to take up external DNA within the cell. So I have already discussed this point. Okay, you can see from this picture the DNA, this particular DNA is outside of the bacterial cell. This one is the bacterial cell. DNA is outside of the cell, and if you treat the bacterial cell with calcium chloride, then the bacteria will be able to take up the DNA within it. Okay. Okay, now next discovery was made by Frederick Sanger, who discovered the method by which DNA can be sequenced. This process of sequencing DNA was discovered by Frederick Sanger in 1977. Now, you know that DNA is nothing but a chain of nucleotides, deoxyribonucleotides. Okay, there are 4 deoxyribonucleotides which are written as T for thymine, A for adenine, G for guanine, and C for cytosine. So all these 4 nucleotides actually remain in random fashion. Okay, like you extend this chain like G, C, T, A, T, D, A, G, C. So thousands or millions of these nucleotides are there in a DNA molecule. So what is the actual sequence of any DNA? That the process of determination of that sequence that I have written here, the nucleotides in this way, that's TAGC, GC, TA, TAG. Okay, so this sequence, how this sequence was determined, that process was discovered by Frederick Sanger. Okay, later I'll discuss in detail about the mechanism behind the sequencing process in a later, in any other video. Okay. Okay, next discovery was made by Kary Mullis in 1985. Discovery was made in 1984, and he published that paper in 1985. He discovered the process by which a small DNA fragment can be amplified. Okay, if a single molecule is there, suppose this is one of the DNA of which you want to amplify this particular portion. Okay, so this particular portion can be amplified in millions of copies. From a single copy, you can produce millions of copies in a biochemical reaction by using a particular enzyme that is known as Taq polymerase, obtained from Thermus aquatica. So this figure represents the detailed procedure, but I'll discuss this procedure in a later video, not in this video. Okay, so just remember that the process of amplifying a small fragment of DNA, that is known as polymerase chain reaction, or PCR, that was discovered by Kary Mullis in 1985. Okay, so this is the PCR machine. This is known as thermal cycler, and these are the small tubes, 200 microliter volume capacity. These are known as PCR tubes, within which 50 microliter of reaction mixture are prepared by mixing DNA molecules, primers, DNTPs, Taq polymerase, and buffer, all those things required for actual reaction to happen. And these tubes are inserted in this particular machine in this place, and is run for 1.5 hours or so, and millions of copies of that particular fragment of DNA you can obtain. Okay, so all these were the major discoveries, I believe, that led to the development of modern biotechnology. Okay, so that's all, and thanks for watching. If you really liked my teaching, please do like this video and subscribe this channel so that we can be encouraged to make more and more videos. Thank you for watching.